Find somewhere comfortable tonight, somewhere your body can be heavy. You don't need to follow this closely. You don't need to remember any of it. If the words wash over you and carry you somewhere quiet, that is exactly what they are here to do. This is not a lecture. This is not something to study. It is simply a place to rest while something gentle and true unfolds around you. If today was hard, you can set it down now. Tonight we are going somewhere very small, smaller than a hand, smaller even than a thought. We are going inside a chrysalis. That jade green casing, no bigger than your thumb, hanging from a leaf somewhere in a garden you may never visit. From the outside, it looks still. It looks like nothing is happening. It looks, honestly, like the most uneventful thing in the world. But inside, something is taking place that has no real equal in all of nature. A living creature is dissolving itself, and from that dissolution, something entirely new is being built. It is, in the quietest and most patient way, one of the most astonishing things that has ever been discovered. And tonight, very gently, we are going to go inside. Make yourself a little more comfortable. Let your shoulders drop. Let your jaw unclench if it has been clenching. Let your hands rest open if they have been holding on. We are in no hurry here. The night is long and soft, and everything that needs to happen will happen in its own time. Long before anyone understood what was happening inside a chrysalis, people simply watched. They watched the caterpillar climb to the top of a stem or curl beneath a leaf and go still. They watched the casing form around it, sometimes green, sometimes brown, sometimes traced with threads of gold. And then they waited. And after days or weeks, something else entirely would emerge from that same small space. Something with wings. For much of human history, this was simply a mystery. A beautiful, quiet mystery that sat at the edge of the knowable world. Aristotle wrote about it. He was endlessly curious about insects, about the way they transformed, about the stages they moved through. He classified and described and wondered. But even he, with all his careful observation, could not see inside. The chrysalis kept its secret. For centuries, the most popular explanation was something called spontaneous generation. The idea that life could simply appear from nothing. That a caterpillar might go into a casing and, through some divine or unknowable alchemy, become a butterfly without any mechanism, without any process, without any thread of continuity between the two forms. It was a satisfying explanation in the way that mystery is always satisfying when you stop asking questions. But in the 17th century, a Dutch microscopist named Jan Swammerdam began asking questions that nobody had thought to ask before. Swammerdam was a remarkable figure, deeply religious, deeply driven, and possessed of a patience that bordered on the otherworldly. He would spend hours, sometimes entire days, bent over his instruments, preparing specimens with a care that his contemporaries found almost incomprehensible. He was working with tools of extraordinary delicacy, and he had developed techniques for dissecting and preserving insects that would not be meaningfully improved upon for generations. And one day Swammerdam did something that changed everything. The wings were already there, already formed in miniature, already present inside the living caterpillar long before the chrysalis had even formed. This was not magic. This was not spontaneous generation. This was a process, an intricate, patient biological process that had been unfolding in secret for hundreds of millions of years inside creatures so small that most people walked past them without a second glance. Swammerdam spent his life documenting what he found. His great work, which he called the Bible of Nature, was a meticulous record of insect life as he had observed it through his lenses. He drew the wings he had found inside caterpillars. He described the stages of metamorphosis with a precision that astonished his readers. He laid the foundation for everything that came after. He died at 43, exhausted and largely unrecognized. His manuscript sat unpublished for decades after his death. But the questions he had asked and the answers he had begun to find did not go away. They waited the way a chrysalis waits. Quiet and patient and full of something extraordinary. To understand what happens inside a chrysalis, it helps to understand what happens just before. A caterpillar spends most of its brief life doing one thing: eating. It hatches from an egg smaller than a pinhead, and it begins immediately to consume. Leaves, mostly. Whatever its species has evolved to eat, it eats with a single-mindedness that is almost meditative in its simplicity. It grows. It sheds its skin. It grows again. It sheds its skin. Some caterpillars molt 4 times before they are done. Some molt 5. Each time they emerge a little larger, a little more substantial, a little more committed to the project of becoming. And then, one day, something changes. A signal moves through the caterpillar's body. A hormonal whisper. The levels of a substance called juvenile hormone, which has been keeping the caterpillar in its caterpillar form, begin to fall. Another hormone called ecdysone rises in response, and the caterpillar, without any conscious decision, without any awareness of what is about to happen to it, begins to behave differently. It stops eating. This is the first sign. A caterpillar that has done almost nothing but eat for weeks simply stops. Then it begins to move, not the slow, purposeful movement of feeding, but something more restless. It walks. It climbs. It seems to be looking for something, though it has no clear idea what. It is looking for a place, a safe place, a hidden place, a place where it can be still for a very long time. When it finds that place, it begins to spin. Not a cocoon, as moths do, wrapping themselves in silk from the outside in. A butterfly caterpillar creates a small silk pad and then a structure called a cremaster, a tiny hook-like anchor, and it attaches itself by this hook and hangs upside down. It dangles there, swaying gently like a small and living mobile. This hanging posture is called a J shape, and it is one of the more quietly extraordinary things you can witness in a garden, if you happen to be looking at the right moment. The caterpillar hangs there for a day, sometimes 2, perfectly still. Committed. And then the final molt begins. The old caterpillar skin splits, starting just behind the head. The creature beneath is not yet a butterfly. It is something in between. Something that has no name in ordinary language. The new skin beneath hardens almost immediately on contact with the air, and it takes on a shape, a color, a form that has nothing to do with the caterpillar. It just was. Within minutes, the transformation of the outer casing is complete. Chrysalis exists. From the outside, all is still. The swaying has stopped. The movement has stopped. Everything has stopped. But inside, in the darkness of that small green shell, something has just begun. Here is the part that seems, at first, impossible. Inside the chrysalis, The caterpillar does not simply rearrange itself. It does not slowly grow wings while everything else stays the same. It does not gradually shift from one form to another the way a clay figure might be reshaped by careful hands. What actually happens is far stranger and far more interesting than that. The caterpillar digests itself. The same enzymes that the caterpillar used throughout its life to break down the leaves it ate are now turned inward. They are released into the body cavity and they begin to dissolve the caterpillar's own tissues. Muscles, organs, the structures that made it a caterpillar, all of it begins to break down into a rich, nutrient-dense liquid. Scientists who have opened chrysalises at this early stage have described the contents as a kind of soup. A biological broth. A fluid that contains all the raw material of the creature that was, ready to be used for something entirely new. This process has a name. It is called histolysis, from the Greek words for tissue and loosening. The loosening of tissue. It is, in its quiet way, one of the most radical acts in all of biology. And yet, it is not chaos. It is not destruction for its own sake. It is an extraordinarily controlled and precise process, regulated by the same hormonal signals that triggered the formation of the chrysalis in the first place. The dissolving is purposeful. The dissolving is, in fact, the first step of construction. Because not everything dissolves. Scattered throughout the caterpillar's body, present since the very earliest days of its life, even since its embryonic stage inside the egg, are small clusters of cells called imaginal discs. The word imaginal comes from imago, the Latin word for the adult form of an insect. These are the cells of the future. They have been there all along, dormant and waiting, surrounded by the caterpillar's tissues, protected from the signals that told the rest of the body to grow and function as a caterpillar. Imaginal discs are, in a sense, blueprints. Each one holds the genetic instructions for a specific adult structure. There is a disc for each wing, a disc for each leg, discs for the compound eyes, for the antennae, for the proboscis, that long coiled drinking straw through which the butterfly will sip nectar from flowers it has not yet seen. These discs are present in the caterpillar, but they are not active. They are patient. They are waiting for their signal. When the caterpillar soup surrounds them, rich with nutrients and hormones, the imaginal discs receive that signal at last. And they begin to grow. This second process, the building, is called histogenesis, the genesis of tissue. Eye cells cluster and multiply into the remarkable compound eye, which will see the world in a way entirely unlike the simple eyes of the caterpillar. Leg cells lengthen and harden. The whole process takes days. For some species in warm summer conditions, the transformation from caterpillar to butterfly can be complete in as few as 5 to 10 days. For species that overwinter inside the chrysalis, it may take months, the whole process slowed by cold to a deep and patient stillness, waiting for spring. All of this happens in the dark, in silence, in a casing no bigger than your little finger. There is something worth sitting with in that image. The idea that the most complete transformation possible, the dissolution of one form and the construction of another, requires stillness and enclosure and time. That the work cannot be done in the open. That it requires a kind of withdrawal. The chrysalis does not apologize for its stillness. It does not perform activity to prove that something is happening. It simply hangs there and trusts the process and waits. The word chrysalis comes from the Greek word chrysos, meaning gold. This is not a metaphor. It is a description. Many chrysalises, particularly those of certain tropical butterfly species, bear genuine metallic gold markings. Not yellowish, not amber. Gold. Bright, reflective, mirror-like gold dots or patches that catch the light in a way that seems entirely out of place on something so small and so quiet. Scientists have various theories about why this might be. Some suggest the gold coloring serves as camouflage, mimicking dewdrops or the reflective patches of light that filter through leaves. Others believe the metallic surface may help regulate temperature. Still others think the markings serve as a warning or as a kind of disorientation for predators that might otherwise recognize the shape as something worth investigating. But whatever the explanation, there is something quietly wonderful about the fact that the word we use for this stage of transformation is rooted in gold. That the ancient Greeks, who could not see inside the chrysalis any more than Aristotle could, chose to name it after the most precious and enduring thing they knew. As if they understood, even without the science, that something valuable was happening in there. Some chrysalises can move. Not all, but many species have a jointed abdominal segment that allows them to twist and twitch when they are touched or disturbed. If you have ever picked up a chrysalis and felt it suddenly writhe in your hand, you will know that this is one of nature's more quietly startling experiences. It is a defense, a way of unsettling a predator or a parasitic wasp that might otherwise decide to lay its eggs inside the casing. The chrysalis cannot run. It cannot hide. But it can, in its small way, resist. And then there is the question of memory. For a long time, the popular understanding of metamorphosis was that everything inside the chrysalis was erased, that the caterpillar dissolved completely, and the butterfly emerged as a clean slate with no connection to the creature that came before. This was a poetic idea. It was also, it turns out, not quite right. In the early 2000s, a researcher named Martha Weiss conducted an experiment that has since become one of the most quietly celebrated findings in insect biology. She and her colleagues took caterpillars of a species of moth and trained them using a gentle conditioning technique. The caterpillars were exposed to a particular smell, and each time they encountered it, they received a very mild electrical stimulus. Nothing harmful, just enough to create an association. After a while, the caterpillars learned to avoid that smell. They had formed a memory. Then the caterpillars pupated. They went through metamorphosis. They dissolved and rebuilt and emerged as adult moths. And the adult moths still avoided that smell. Not all of them. Not perfectly. But a significant number of them, when presented with the odor they had learned as caterpillars, still showed signs of avoidance. The memory had survived. It had crossed the boundary of dissolution and rebuilding and arrived intact on the other side. This finding raised questions that researchers are still working to answer. Some structures, particularly those associated with the brain's deeper architecture, appear to survive the process, carrying with them a kind of experiential residue from the larval stage. It is worth noting that this is not universal. Fruit flies, which also undergo complete metamorphosis, appear to experience much more substantial brain remodeling, and their larval memories do not seem to persist in the same way. The nervous system of each species navigates metamorphosis according to its own biology, but for certain moths and butterflies, something of the caterpillar's experience endures. A thread of continuity runs through the dissolution, invisible but real, connecting the creature that was to the creature that becomes. There is something in that which feels important, though it is hard to say exactly why. The idea that transformation does not require forgetting. that you can become something entirely new and still carry something of what you were, that the dissolution is not an erasure. Let us go a little deeper into the machinery of this, because it is genuinely beautiful once you see how it works. The entire process of metamorphosis is orchestrated by hormones, chemical signals, vanishingly small in quantity, that travel through the insect's body and tell its cells what to do and when to do it. The precision of this system is extraordinary. A tiny shift in the concentration of a single hormone can mean the difference between a caterpillar that continues to grow and one that begins the process of transformation. The key players are 3 in number. The first is juvenile hormone. This is the hormone of continuation, the hormone that says, stay as you are. As long as juvenile hormone is present in sufficient quantities in the caterpillar's blood, each molt produces another caterpillar, larger but still a caterpillar. The imaginal discs remain dormant. The wings remain folded and waiting. The caterpillar continues its work of eating and growing. The second is a hormone called ecdysone, sometimes called the molting hormone. Ecdysone is produced by a small gland in the caterpillar's thorax, and it is what actually triggers each molt. Without ecdysone, the old skin does not split. Without ecdysone, the caterpillar cannot grow. The third is a signaling molecule called prothoracic atropic hormone, which is produced by cells in the caterpillar's brain and tells the thoracic gland to produce ecdysone. The brain, in other words, is involved from the very beginning. Even in a caterpillar, even in a creature with a nervous system far simpler than our own, the brain is part of the conversation. When the caterpillar has eaten enough, when it has grown through its final larval stage, something changes in the balance of these signals. Juvenile hormone levels fall below a threshold. The imaginal discs, which have been suppressed by its presence, begin to respond to ecdysone in a new way. Instead of triggering another larval molt, the signal now triggers the formation of the pupa. The chrysalis forms. The transformation begins. In the last few years, researchers have identified another layer to this system. A gene called E93 appears to be a critical switch in the metamorphic process. Scientists at the Institute of Evolutionary Biology in Barcelona have shown that E93 expression plays a central role in initiating the adult developmental program. It is not simply that juvenile hormone disappears and everything else follows automatically. There is a positive signal, a genetic instruction that says, now build the adult. E93 appears to be a significant part of that instruction. This kind of research, patient and incremental and conducted in laboratories that most people will never visit, is slowly illuminating the molecular architecture of one of biology's oldest and most beautiful processes. Xavier Bells, who has spent decades studying insect metamorphosis, published a comprehensive synthesis of 30 years of research in 2020, drawing together the threads of hormonal and genetic regulation into a picture of remarkable coherence and complexity. And yet, there is still much that is not fully understood. The precise way in which juvenile hormone acts at the molecular level remains an active area of investigation. The mechanisms by which certain memories survive the dissolution of the nervous system are still being mapped. Science, like metamorphosis, does not complete itself all at once. It proceeds in stages, each one building on what came before, each one revealing new questions nested inside the answers. There is a patience required in that kind of work that feels, somehow, related to the patience of the chrysalis itself. The willingness to stay with something incomplete, to trust that the process is moving even when nothing appears to be happening from the outside. The caterpillar does not know what it is becoming. It cannot know. It has no concept of flight, no memory of having seen a butterfly, no way to imagine the compound eyes or the proboscis or the wings that are, even now, assembling themselves from the soup of its dissolved tissues. It simply surrenders to the process. It hangs upside down in the dark, and it trusts. There is a connection worth resting with as the night grows deeper around you. What the chrysalis does in its small and patient way is not so different from what your brain does when you sleep. This is not a metaphor chosen for its prettiness. There is something genuinely true in it. When you close your eyes tonight, when the last of the day's alertness finally fades and your breathing slows and your muscles release their long-held tension, your brain does not simply go quiet. It enters a different kind of activity. An inward activity, a reorganizing, dissolving, rebuilding kind of activity. That is, in its own way, just as extraordinary as what happens inside a chrysalis. In the earliest stage of sleep, your brain's electrical activity slows from the busy, jagged rhythms of waking life into something gentler. The fast, high-frequency waves of alert attention give way to slower, more synchronized patterns. Alpha waves, which move at something between 8 and 12 cycles per second, replace the urgent chatter of full wakefulness. Then, as sleep deepens, even these give way to the slower undulations of theta waves, rolling through your brain like long swells on a deep ocean. And then, if you sink far enough, something remarkable happens. Your brain enters what sleep researchers call slow-wave sleep, the deepest and most restorative stage of the night. In this state, your brain produces what are called delta waves, vast and slow electrical rhythms that move through the sleeping brain at less than 4 cycles per second. These are the slowest brain waves we produce. They are the signature of the deepest rest. During slow-wave sleep, something is happening to your memories. The experiences of the day, the things you learned and felt and encountered, are being sorted and processed and integrated into the larger architecture of what you know. Researchers at the Paris Brain Institute have found that even in deep sleep, certain faster wave patterns, including beta and gamma rhythms associated with inhibitory neurons, appear to replay patterns from the previous day's experience, as if the brain, in its deepest rest, is quietly rehearsing what it has learned, weaving the new into the old, building something coherent out of the day's raw material. This is not so different from the imaginal discs, those tiny clusters of cells that carry the blueprint of the adult butterfly and wait, through all the dissolution, to begin their work of building. Your memories have their own kind of blueprint. The traces left in your neural networks by the day's experiences are waiting, in the quiet of sleep, to be consolidated, to be made permanent, to be woven into the larger structure of who you are. And in REM sleep, the stage where your eyes move beneath their closed lids and your brain produces waves not unlike those of wakefulness, something else is happening. Emotional memories are being processed, complex skills are being integrated. The strange associative logic of dreams may be part of the brain's way of finding unexpected connections between things it has learned, linking distant pieces of experience in ways that would not be possible in the linear, sequential thinking of waking life. Perhaps dreaming is a little like that, a different kind of seeing. There is also a more literal kind of cleaning that happens during sleep. The brain has a system called the lymphatic system, a network of channels through which cerebrospinal fluid flows during sleep, washing away the metabolic byproducts that accumulate during the day. The proteins and waste products that, if they were allowed to build up, would slowly compromise the brain's function. Sleep clears them away. Sleep maintains the architecture. The chrysalis, in its own way, does the same thing. The soup of dissolved caterpillar tissues is not discarded. It is recycled. Every molecule of energy and protein that the caterpillar spent its life accumulating is used in the construction of the butterfly. Nothing is wasted. The dissolution is not destruction. It is preparation. When you are exhausted, when your body is heavy and your thoughts are slow and you cannot quite hold on to what you were trying to think, that is not failure. That is your body asking, in the only way it knows, for the time it needs to do its work. The caterpillar does not fight the chrysalis. It does not resist the stillness. It surrenders to it, and in that surrender, something extraordinary becomes possible. If today was heavy, if you are carrying something you have been carrying for a while now, if the world has felt like too much and your own mind has felt like too much, you are allowed to set it down here. Not forever. Just for tonight. Just for the hours of darkness that are already quietly gathering around you. The imaginal discs do not rush. They do not know about deadlines or expectations or the accumulated weight of everything that needs doing. They simply begin, when the conditions are right, to build what they were always meant to build. You have your own version of those blueprints. The deep, unhurried knowledge of who you are beneath the surface of everything that has happened to you. Sleep is one of the ways that knowledge gets to do its quiet work. Insects that undergo complete metamorphosis, the butterflies and moths and beetles and bees, account for nearly 60% of all known animal species, more than 1 million described species, possibly tens of millions more that have not yet been found and named. This way of being, this strategy of dissolving and rebuilding, of withdrawing from the world to undergo transformation before returning to it in a new form, is not a rare or fragile thing. It is one of the most successful strategies life has ever discovered. There may be something in that worth holding on to, not as a grand claim, but as a quiet reassurance. The withdrawal is not weakness, the stillness is not stagnation. The dark, enclosed, apparently uneventful space of rest is where some of the most important work gets done. Somewhere tonight, in a garden or a hedgerow or a woodland edge, a chrysalis is hanging in the dark. It has been there for days, perhaps, or weeks. It is utterly still. To a passing bird or a hurrying human, it looks like nothing. It looks like the absence of activity. But inside, in the patient dark, wings are assembling themselves from the dissolved remnants of a creature that no longer exists. Compound eyes are forming that will see the world in colors invisible to us. A proboscis is coiling itself into a perfect spiral, ready to unroll into the heart of a flower it has not yet imagined. All of it happening in silence, all of it happening in stillness, all of it taking exactly as long as it needs to take. The night is deep around you now. Whatever you are carrying when you lay down, let it rest where it has fallen. The chrysalis does not carry the caterpillar into the butterfly. It lets it go. It uses what it needs, and it lets the rest dissolve into something new. You have done enough today. Whatever that means for you, whatever shape your day took, you have done enough. The work of tonight is quieter. The work of tonight is the kind that happens when you stop trying. Somewhere in the dark, something small and green is hanging from a leaf, doing nothing, becoming everything. Well, There is a caterpillar species native to the forests of eastern North America, the spicebush swallowtail, whose final larval stage is a remarkable thing to observe. As it prepares to pupate, its body shifts color from green to a deep, rich orange-yellow, a change triggered by the same hormonal cascade that is already beginning to move through its tissues. The color change serves as a kind of announcement, not to us, but perhaps to the caterpillar's own biology. A visible signal that the transition is already underway, already written into the flesh before the chrysalis has even begun to form. This detail, small as it is, carries something worth noticing. The transformation does not begin when the chrysalis forms. It begins earlier, quietly, in ways that are not yet visible from the outside. The outer form may still look like a caterpillar. It may still be moving, still hanging, still recognizably itself. But something has already shifted inside. The decision, if we can call it that, has already been made. There are caterpillars that travel remarkable distances in search of the right place to pupate. For a creature whose body is perhaps 5 centimeters long, that is an almost incomprehensible journey. And yet it makes it, driven by nothing more than a chemical signal and something that functions in its way like instinct. It does not know where it is going. It simply goes. It trusts that the place will be found. When it finally stops, when it settles into the corner of a wall or beneath a loose piece of bark or deep in the tangled roots of a hedgerow and begins to spin the loose cocoon that will house its pupa, there is something in that arrival that feels like relief. Even in a creature without the capacity for relief as we understand it, the end of the walking has a quality of completion to it. The searching is over. The stillness can begin. The soup stage of metamorphosis, that liquid interior state in which the caterpillar's tissues have broken down, but the butterfly's structures have not yet fully formed, lasts for different durations in different species. In a monarch butterfly, the whole process of dissolution and rebuilding takes roughly 10 days under warm conditions. In a morning cloak butterfly, which overwinters as an adult and whose chrysalis stage is relatively brief, the transformation can be substantially faster. In certain species that diapause, that enter a kind of suspended developmental state during cold months, the chrysalis may persist for the better part of a year. The processes inside slowed almost to a halt by cold, waiting for the warmth that will restart them. Researchers who have studied the internal chemistry of the chrysalis at different stages have found that the timing of histolysis and histogenesis is extraordinarily precise. The dissolution does not simply continue until everything is gone. It reaches a point, a kind of minimum, a nadir of structure, and then the building begins to outpace the dissolving. The imaginal discs, which have been growing slowly and quietly even as the surrounding tissues break down, begin to accelerate. The system tips. The balance shifts from undoing to making. One of the more remarkable aspects of the imaginal disc system is how early these structures are established. In many butterfly species, the imaginal discs are present in the embryo before the caterpillar has even hatched from its egg. They are laid down during embryonic development as small groups of undifferentiated cells set aside from the rest of the developing body. They do not participate in the caterpillar's life in any direct way. They do not contribute to its digestion or its movement or its growth. They simply exist, waiting, surrounded by the caterpillar's tissues, maintained but not activated, preserved through every molt, through every growth phase, through the entire long project of being a caterpillar, until the moment their signal finally arrives. There is a geneticist named Carroll, Sean B. Carroll, who has written with great clarity about the imaginal disc system and what it reveals about the deep logic of development. He describes the imaginal discs as a kind of parallel program running silently inside the caterpillar. A second set of instructions that the caterpillar carries without using. A future written into the present that the present cannot yet read. The caterpillar, in his framing, is in some sense two creatures simultaneously. The creature it is, and the creature it is becoming. Both programs running at once, one dominant and active, one dormant and waiting. This image of two programs running in parallel, of a future self quietly present inside the current self, has stayed with researchers and writers who have encountered it in ways that go beyond the strictly scientific. It is the kind of idea that arrives gently and then remains, settling into the mind like a stone settling into still water. The metallic gold markings found on certain chrysalises are among the more visually striking things in the insect world, and they have attracted genuine scientific curiosity precisely because they seem so improbable. Gold, as a color in living organisms, is exceptionally rare. Most biological colors are produced by pigments or by structural interference, the way a butterfly wing produces iridescent blues by the physical arrangement of microscopic scales, rather than by any colored dye. But the gold of a chrysalis is something different again. It appears to be produced by a combination of structural reflection and underlying pigmentation that creates a genuinely metallic appearance, one that changes slightly with viewing angle, that catches the light and releases it in a way that seems almost deliberate. The chrysalis of the Gulf fritillary butterfly, a species found across the southern United States and Central America, bears some of the most striking gold markings of any North American species. Small, bright, perfectly placed dots that run along the dorsal surface of the pupal case like a constellation. Researchers studying their optical properties have found that these spots are genuinely reflective in the way that polished metal is reflective, not merely shiny in the diffuse way of many insect surfaces. The mechanism behind this remains an area of active interest. There is a related phenomenon in the chrysalises of certain heliconide butterflies, the passion vine butterflies of tropical America, whose pupae can take on an almost mirror-like silver sheen over their entire surface. These species have been studied extensively, partly because of their remarkable wing patterns and their role in mimicry complexes, and the optical properties of their chrysalises have been examined with considerable care. The reflective surface appears to function partly as camouflage, the mirror-like quality rendering the chrysalis nearly invisible against the shifting, dappled light of the forest floor or the underside of leaves, reflecting its surroundings back at any observer rather than presenting a solid, recognizable form. The ancient Greek decision to name this stage after gold, before any of this was understood, before microscopes or spectrophotometers or the science of structural color, was made purely on the basis of looking, of paying attention, of finding something so beautiful and so strange that the most precious word available seemed somehow appropriate. There is a quality of attention in that which feels worth honoring. The careful, unhurried attention of someone who has noticed that the world, looked at closely enough, gives back remarkable things. The question of memory surviving metamorphosis opened doors that researchers had not anticipated. Following Martha Weiss's work with caterpillars and moths, other researchers began to look more carefully at what exactly persists through the pupal stage. Studies using increasingly sophisticated imaging techniques have begun to map the neurological architecture of the larval and adult brain in species like Manduca sexta, the tobacco hornworm moth, whose relatively large nervous system makes it a useful model for this kind of work. What these studies have found is that the metamorphic process is not, in fact, a wholesale demolition and reconstruction of the nervous system. Certain neurons that are present in the larval brain persist into the adult. They change. They extend new connections. They retract old ones. They find themselves in a new chemical environment with new neighbors and new functional roles. But they survive. The cells themselves cross the boundary. And with them, it appears, certain functional properties survive as well. The patterns of connectivity that encoded a learned association in the caterpillar may be preserved, altered but recognizable in the adult moth. This has led some researchers to describe metamorphosis not as a death and rebirth, but as a radical renovation. The building is transformed beyond recognition. Its function changes entirely. But some of the original structure remains within the new walls, carrying traces of the history of the place. The work of Xavier Bells and his colleagues in Barcelona is part of a broader international effort to understand metamorphosis at the genetic level, an effort that has accelerated considerably in the era of genomic sequencing. The ability to read the entire genome of an insect, to track which genes are active at which stages of development, to identify the precise molecular targets of hormones like juvenile hormone and ecdysone, has transformed the field in ways that would have seemed extraordinary even 20 years ago. One of the findings that has emerged from this work is that the genes involved in insect metamorphosis are, in many cases, ancient. They are not novel inventions that appeared with the first holometabolous insects, those insects that undergo complete metamorphosis roughly 300 million years ago. Many of them are deeply conserved across the animal kingdom, present in species that do not metamorphose at all, doing other jobs in other contexts, recruited by evolution into the metamorphic process from a toolkit that was already old when insects were young. This is one of evolution's recurring strategies. Not invention from nothing, but repurposing. Taking what is already there and finding a new use for it. The imaginal disk system itself may have evolved from simpler developmental structures present in the ancestors of modern insects. The hormonal cascade that orchestrates metamorphosis uses signaling molecules that, in other animals, serve other functions. The whole extraordinary process is built at its molecular foundation from borrowed parts. There is something quietly encouraging in that. The most transformative process in the animal kingdom is not built from unprecedented raw material. It is built from what was already available, organized in a new way, pointed toward a new purpose. The period just before emergence is called ferret, from the Greek word for mantle or covering. The Faraday butterfly exists fully formed inside the chrysalis, complete in every detail, wings folded against its body, legs and antennae arranged in the careful geometry of the pupa, waiting. The transformation is complete. But the butterfly has not yet emerged. It is dressed in its old case, wearing the chrysalis as a final garment, waiting for the right conditions. In many species, emergence is triggered by light. The first light of morning, falling on the chrysalis after a night of cooling, sends a signal through the case to the waiting butterfly inside. This is why, if you have ever found a chrysalis and brought it indoors, the butterfly that emerges may do so at an unexpected time. It is responding to the light in the room, which does not follow the same rhythms as the light outside. The biology is precise. The timing is important. A butterfly that emerges in the middle of the night, in cold conditions, before its wings have had time to expand and dry in warm air, may not survive. The act of emergence itself is remarkably brief. What took days or weeks to build takes minutes to reveal. The chrysalis casing splits along the seam that was built into it from the beginning, a line of weakness that exists for exactly this purpose. The butterfly pushes its way through, legs first, then head, then the crumpled wet mass of its wings. It clings to the empty case or to whatever surface is nearby, and it pumps fluid from its body into the wing veins. The wings expand, slowly, from a crumpled pad into the full, symmetrical, extraordinary structure they were always going to be. The fluid drains back into the body as the wings harden. The proboscis, which emerged in two halves, is carefully zipped together by the butterfly using its legs, forming the single coiled tube it will use for the rest of its life. All of this takes perhaps an hour. Then the butterfly is still for a time, wings held open or folded, drying in the air. And then, when the wings are ready, it flies. The first flight of a butterfly is not tentative. It is not a stumbling, uncertain thing. The butterfly that has never flown before takes to the air with a competence that seems, from the outside, impossible. The neural circuits for flight were built into it during metamorphosis. The muscle coordination, the wing geometry, the sensory feedback loops that allow it to adjust its path in real time, all of it was assembled in the dark, without practice, without rehearsal, without any previous experience of moving through air. And yet, it works. The first time, it works. This is, perhaps, the most astonishing thing about the whole process. That you can build something so complex in the dark without testing it and have it function perfectly the first time it is needed. The glymphatic system, that slow river of cerebrospinal fluid that moves through the sleeping brain, was only described in detail relatively recently. A researcher named Macon Nedegard at the University of Rochester, working with colleagues in the early 2010s, published findings that illuminated this system in a new way, showing that the channels through which the fluid flows actually widen during sleep, allowing a significantly greater flow of fluid through the brain's tissue than occurs during waking hours. The brain, it appears, physically opens itself to cleaning when it sleeps. The architecture shifts to allow the maintenance to happen. Nedegard described it, in conversation with journalists, as something like a dishwasher, which is perhaps a slightly more prosaic image than the Chrysalis. But the underlying logic is the same. The system cannot clean itself while it is running at full capacity. The cleaning requires a different state, a slower, more inward state, a state that looks from the outside like nothing much at all. The proteins that the glymphatic system removes during sleep include amyloid beta, a substance whose accumulation in the brain is associated with certain neurodegenerative conditions. Sleep, in other words, is not merely restorative in the loose general sense. It is specifically protective. It is doing targeted work that waking cannot do. The apparent absence of activity is, in fact, the activity. When you have not slept enough, the world is different. Not just slower, not just foggier, but genuinely different. Colors are less vivid. Sounds are more irritating. Small frustrations become large ones. The emotional processing that sleep normally performs has been interrupted, and the raw material of the day's experiences sits unprocessed, closer to the surface, more reactive, more tender. This is not weakness. This is simply what happens when a process that needs to run is prevented from running. The chrysalis, interrupted, cannot complete its work. There is a particular kind of tired that goes beyond the body. The tiredness that comes from carrying something difficult for a long time. From navigating a world that asks more than you have to give. From the accumulated weight of small disappointments and large uncertainties and the relentless forward motion of days that do not pause to ask how you are doing. This kind of tiredness does not always resolve in a single night. Sometimes the chrysalis needs longer than expected. Sometimes the cold of a difficult season slows everything down and the waiting extends beyond what was anticipated. The morning cloak butterfly, which overwinters as an adult, sometimes emerges on warm days in late winter, only to retreat again when the temperature drops. It does not force the spring. It waits for the conditions to be genuinely ready. You are allowed to take the time that you need. This is not a small or easy thing to believe in a world that values speed and output and visible productivity above most other things. But the biology is clear. The rest is not separate from the work. The rest is part of the work. The dissolution is part of the construction. The dark is part of the becoming. Somewhere in the long history of life on this planet, the first insect to undergo complete metamorphosis did so without knowing what it was doing. It dissolved and rebuilt according to instructions written into its genome by millions of years of slow refinement. Instructions that have been tested and adjusted and passed down through uncountable generations, each one contributing a small increment of precision to the process. The result, accumulated over hundreds of millions of years, is one of the most elegant and successful biological strategies ever to have existed. Your own capacity for rest and repair is equally ancient. The mechanisms of sleep, the hormonal rhythms of the circadian cycle, the architecture of slow wave and REM sleep, the glymphatic cleaning system, all of it is the product of an evolutionary history that stretches back far beyond our own species. You are not doing something new when you sleep. You are doing something very, very old. Something that has been refined by time into a precision that we are only beginning to understand. The night around you is the same night that has always been there. Patient and vast and unhurried, it has held every sleeping creature that has ever lived, every chrysalis that has ever hung in the dark, every exhausted body that has ever finally, gratefully let go of the day. It holds you now,